And whenever you would like to start, we can go.
Great. Well, I'll go ahead and introduce Meigan. Meigan is an undergraduate at Michigan State. She took the iBio 150 class that I teach, which is really a course for largely engineering and computer science majors. And then she approached me after the class wanting to know if she could do a professorial assistantship. Meigan is a computer science major, so I was really excited that she was interested in biology. And so she has been working with Danielle and I, working with Danielle and I on sort of an extension of the salamander research project that I'm involved with through SparkNet. So it's really great to have Meigan presenting, and she's an amazing undergraduate, and I'm happy that she's going to be coming back next year, and hopefully we can draw her over to the dark side and convince her to continue on to a PhD, but she's doing great. So anyways, with that, Meigan, I'm going to turn it over to you. Thank you for sharing all of this work with us.
All right, great, thank you. So as Dr. Mead was saying, my name is Meigan, and I've been working with her as well as Dr. Whitaker on our project, and we like to call it—
Sorry.
So we call this our project Salamander Scents, and with that, we've been looking at the potential for communication through volatile compounds in the redback salamander. So we've been looking at analyzing the volatile compounds and the microbiome associated with the redback salamander. So just to give like an introduction, as I said, we've been working with the eastern redback salamander called Plethodon cinereus, and they are a terrestrial lungless salamander, very common across like all of the eastern US, as well as in parts of Canada. And then they also come in two color morphs, which you can like see here in this photo. So there's the striped morph, which is like a gray color with a red stripe on the back, and then the unstriped or leadback morph, which is just the gray color without any red markings or anything like that. And then these salamanders are very commonly studied in the field of ecology because they're very abundant within forested areas in the eastern US, and then also because they're often noted to be like a keystone species in many of these forests. So just kind of moving on into studies that are more like related to how we've been working with communication and looking at those with the salamanders. So it's been shown a lot that the salamanders do have the potential for conspecific communication with each other. Specifically, a lot of studies have been looking at like the territories that these salamanders hold. It's been shown that adults, both males and females, are holding territories for both mate selection as well as just food and other resources. And with that, they have been shown that they potentially recognize kin, largely because they've been shown to have more like aggressive behavior against unfamiliar salamanders. And with these studies, a lot of them have been using just chemical cues, and those have shown that the salamanders are reacting to chemical cues and not just like visual cues of what the salamanders look like. And they haven't been able to identify exactly what the salamanders, what information the salamanders are gaining from those chemical cues, but it has been hypothesized that they are at least able to determine the sex of salamanders. And this is because like they've noticed the aggressive behavior is really common towards same-sex salamanders. Like male salamanders are very aggressive against like unfamiliar male salamanders, and female salamanders are more aggressive against other female unfamiliar salamanders as well. And so with holding territories, it's also noted that they are likely communicating what the boundaries of their territories are. And there are two kind of ways that researchers have seen this happen. So the first is through granular secretions, most notably through the postcloacal gland. So the cloaca is just the end of their urinary tract, digestive tract, and reproductive tract, and it's located just right under the base of their tail. And it's been noted that they have like a lot of glandular activity there. And they also have a behavior that is known as like the postcloacal press, where it's been seen that the salamanders will like rub their cloaca against different areas in the environment. And this is hypothesized that it's done to mark their territory with these secretions from the cloacal glands. And then in addition to that, they've also been noted to use like fecal pellets a lot for this communication. And again, a weak line of defense for marking their territories. So especially with females, it's been known that they'll like do fecal pellet squashing. So they'll like put their nares in the fecal pellet and squash it. And a lot of the research hypothesizes this is for like mate selection, trying to find other male salamanders that have a really high diet, a really highly nutritious diet, but there's a lot of potential that they're also gaining other information about the male as well, such as like their potential like ID, again, for kin recognition and other statuses that they may have. And then looking into like the microbiome side of the studies with the salamanders, a lot of the studies with the bacteria and looking at their skin have been focused on antifungal properties of bacteria. So currently there's a really big fungus around called the chytrid fungus that has been infecting a lot of different amphibians all around the world. So a lot of research has focused on amphibians, including the redback salamander, and looking if they may be resistant to or immune to different fungal infections because of the bacteria on their skin. And so one of the studies we focused a lot on was by Loudon and his team from 2014. And in that study, they found what's called what they called a core community of bacteria. And they found this core community by dividing up all the information that they got, and they divided the bacteria into 8 different operational taxonomic units, or OTUs, that were found across over 90% of the salamanders that they had in their sample. So with that, they found that this core community, of course, was associated with all of their salamanders, but was also really impacted by the environment. Notably, they found that all of the bacteria that were associated and found on the salamanders were also found in the environment that they're living on. So they believe that what the salamanders are gaining the bacteria from the environment, but their skin is actively selecting for different traits in this bacteria, such as like antifungal properties. And so with that, they've also shown that the environment kind of acts as a reservoir of bacteria, and that living in an environment helps to keep the core community regulated and increase the diversity of bacteria on the skin. So on this graphic that I've shown here from their paper, they housed salamanders in 2 different medias. So the first was a sterile media, and then the second was a soil environment from where they were captured in the field. And they found that the salamanders living in that sterile media, the core community really just dominated their microbiome, and they had a very low diversity within their microbiome, versus salamanders that lived in the natural soil environment had a more regulated core community with a higher diversity level in their microbiome. And then additionally, the salamanders, kind of going with the communication and the microbiome, they've been studied a lot for their pheromones. It's been really well noted that they use 3 different pheromones during courtship. And so these are the plethodontid modulating factor, the plethodontid receptivity factor, and then the sodepherin precursor-like factor. And all of these are used to help make the females slow down or more receptive to courtship. So during courtship, the males use what's called their mental gland, and that's located right under their chin right here. And so what they'll do is the males will slap their mental gland against the nares of the female in order to deliver those pheromones, and again, make her more receptive to courtship and mating with them. And so interestingly, with these pheromones, they've been studied a lot, but they've only been found to be associated only with the mental gland. So specifically, Catherine Palmer, she's been looking a lot at these pheromones and has found that they're not found anywhere outside of the mental gland. So they looked at different swabs, different areas on the salamander, and only were able to find them in the mental gland. In addition to that, these pheromones are found a lot in the other related species to redback salamanders, like in Plethodon shermani, which is in the picture. And they found that these pheromones across all these species are exhibiting a very high sequence variation. And so since all the studies have shown that they're using pheromones for courtship, it's just been assumed that they also use pheromones for other forms of communication. And so this is kind of one of the reasons we wanted to look at the potential for other forms of communication, specifically volatile communication in these salamanders, just because there's kind of question marks with all this high sequence variation and they're only found in the mental gland. We're kind of wondering how they're able to communicate outside of courtship. And again, if they aren't using just the assumed pheromones. And then just to go along with that, their nasal cavity anatomy does allow them to be able to pick up and respond to volatile cues in the environment. So specifically in their nasal cavity, they have 2 different organs. So the verminal nasal organ, which is responsible for receiving and interpreting non-volatile cues, and then the other organ, which is used for receiving and interpreting volatile cues. And then lastly, just kind of informing our hypothesis that salamanders may be communicating in other ways outside of pheromones, is that this has been shown in a lot of other species that symbiotic bacteria are creating volatile compounds that help the other animals communicate. So of course, one of the big ones is the dark-eyed junco, which Dr. Whitaker has been working very closely on. They found that the prenoil of the juncos have these bacteria that produce compounds that the birds are using as social cues. And then additionally, it's been found in quite a few mammals as well, specifically hyenas and Bengal cats. With the mammals specifically, they look a lot into what's called the fermentation hypothesis. And with that, it's just that they believe that, again, these symbiotic bacteria produce these volatile compounds that are used for communicating with other animals in their species, and that the difference in all these compounds in the social cues is actually due to variety or difference in the microbiome itself. And so specifically with hyenas, they found that their scent glands are the one responsible for housing the symbiotic bacteria, and then with Bengal cats, their angle sac secretions have these as well. So kind of moving on to what we've already done so far on this project, I just want to kind of iterate that this is a really first step. So we have a lot more to go through, but we've so far been able to do quite a lot, and which we're excited about. So we took 2 different sample sites and we were able to gather samples of salamanders in fall of 2020. So we looked at 2 different areas around MSU, so Rose Lake and Toomey Woodlot, which you can kind of see where they are located, related to each other in the map here. And so for our initial sampling of the environment, we went out and turned over rocks and logs and things, common places where the salamanders may live, looking for adults as well as juveniles that we could sample. And then just on additional samplings, we actually decided to use cover boards instead, just so we were able to get more consistent counts of salamanders and get as many swabs as possible to get a really large sample size. And of course, after we captured the salamanders, we recorded all of their characteristics, such as their sex, their age, where they were captured, as well as the color morph that they were. And so we decided to swab 3 different areas of the salamanders. So the first place we decided to target was the mental gland. Again, that's located under the chin of the male salamanders, but even for the females, we also swabbed the area just to kind of compare what maybe the male mental gland looks like versus what the female's non-mental gland chin area looks like. We also decided to swab the cloaca, again, because of that importance with those post-cloacal gland secretions, as well as that's where the fecal pellets are deposited from. And then we also decided to swab the ventral side of the salamanders, kind of to act more as a neutral control-like area, because that's not really associated so far with any of their major communication. And then we also housed these salamanders for a very short period of time and waited for them to deposit fecal pellets. And so if the salamanders did deposit fecal pellets, we went ahead and swabbed those as well. So each of these 3 areas, as well as the fecal pellets, were swabbed twice, once for a microbial analysis and then once for a volatile compound analysis. So looking just briefly into these analyses that we have done. So for the volatile analysis, we've been working with the Research Technology Support Facility here at MSU, and we used gas chromatography mass spectrometry for the identification of those compounds using an internal standard. And we've been working with the software Mass Profiler Pro, as well as R and R Studio, to identify those compounds and to do some really basic data analysis of the volatiles. And then for the microbiome analysis, this is more in progress right now. We've just finished up the DNA extractions using just a basic QIAGEN kit. And moving forward, we plan to sequence the V4 region of the 16S RNA in order to identify the different bacteria that we're having in the salamander skin. And when we get those sequences done, we plan to do all of our sequence analysis using Mother, ideally kind of grouping what we find into different operational taxonomic units, again, identifying how they may be different between individual salamanders or different body sites and areas and things. So looking at just kind of the basic results that we've gotten to so far, the first thing we've done is look at the most abundant volatile compounds. So after the GCMS, we were able to find the peak areas of all of our compounds, and we combined all of those into our 30 most common peak areas, the ones that had the largest areas that indicate that they were the most abundant volatile compounds across all of our individual salamanders. And then from that, we threw out anything that couldn't be identified for sure, as well as any sort of contaminants that may be in those volatiles. And so this is the list that we've come up with so far. Moving forward, we really want to kind of look more into all of these compounds and kind of see if any of them have already been associated with communication in birds or mammals so far. And then just also some other results looking at our basic volatile data. Again, this was all done using R. So the first thing that we've done was a non-parametric multivariate ANOVA test using all of our samples. And to do this, we just wanted to see kind of how individual volatile profiles may be differing from the different body sites of our different volatile profiles. And so far we found a really interesting trend where individuals are differing, but body sites are not. So as you can see here on this plot, the markers that are the same color are clustering a little bit more closely together versus markers of the same shape. So this is indicating that swabs from the same individual salamanders are more closely related than swabs of the same body site. So for example, like a cloacal swab from salamander 1 is more similar to a mental gland swab to salamander 1 versus the cloacal swab from salamander 2. So obviously we can't really say anything definitive here, but we are really excited about this result because it does potentially indicate there might be some sort of like kin recognition going on, or that individuals might have their own specific like volatile profiles that other salamanders can like read in order to gain information about that individual. And then moving on, we've also done a non-parametric multivariate ANOVA test, this time using one sample for every individual. So as I just mentioned, we found that there are no differences between the body sites. So we decided to choose one body site and then look at that body site across all the different individuals to see how our other factors that we've been looking at might be affecting the volatile profiles of each of the salamanders we've been looking at. So we found that we had 11 salamanders from Toohey Woodlot and 9 salamanders from Rose Lake. And we found that localities are differing across the board, whereas sexes, ages, and morphs are not differing. So this means that salamanders from Rose Lake are most similar to other salamanders from Rose Lake versus salamanders from Toohey Woodlot, and that salamanders from the same location are more closely related to each other because of that location versus than because of their sex, their age, or their color morph. And so interestingly, we did find this locality difference trend in both the mental, when we separated by the mental swab and the ventral swab, but not by the cloacal samples. And we think this might just be because of our small sample size, since it was kind of on like the threshold of significance, but we're looking to kind of increase our sample size and see if that trend continues on moving forward. So just kind of looking more at each of the different characteristics that we've been looking at more closely. So first here looking at the locality and the age, we found 7 juvenile salamanders and then 13 adult salamanders. And something that we really is important for our studies that all of 7 of our juveniles were found from Toohey Woodlot. So we want to go in the future, go back to Toohey and try and get more adult salamanders, because this juvenile number might be definitely affecting that the age significance or non-significance in this case, as well as the locality significance. So we want to see if getting more adults and comparing like adult juveniles from Rose Lake and adults from Rose Lake and adults from Toohey Woodlot, if this trend still holds where the locality is like that driving factor in what's making those volatile compound profiles different. And then we also, of course, looked at the locality and the sex of the salamanders. We had 6 female salamanders and 4 male salamanders, and then 10 unknown, and those were just juvenile salamanders or adults that could not be sexed correctly. And so again, we find this trend where the locality is that driving factor. So in these plots, like all those markers of the same shape are clustering really closely together, whereas the colors are kind of all over the place, indicating that sex really isn't what's determining all these different volatile profiles that we're looking at. And again, because of that large number of unknown, mostly juvenile salamanders, we want to go forward and kind of see if we can get more samples to see if we get new trends or if this trend continues to hold. And then lastly, we looked at the locality and the color morph. So we found 16 striped salamanders and then 4 of the leadbacks. And again, we kind of have a really unbalanced sample size here. That's just because here in Michigan, we tend to get a lot more of those striped salamanders instead of the leadbacks. So we're going to try and get some more leadbacks, but that's not always the case. We kind of just want a larger sample size as possible. But we are interested in seeing that if maybe we did have a more even sample number, if maybe morph would be one of those driving factors that might be changing the volatile profiles, because that is unique to each individual salamander. And then just to wrap up, again, this is a very first preliminary step that we've done so far, but we're really excited about these trends that we've seen already, that differences in the individuals and of course that difference in locality as well. So for some short-term goals that we have kind of moving over into next year, and I guess the end of this year, we want to finish up the microbiome analysis as much as we can to get our sequencing done and then start working in Mother to group all of our things into operational taxonomic units, really find what kind of bacteria is living on different individuals and then different body sites. And again, kind of do a very preliminary preliminary analysis of that and look again if we see these same trends that we're seeing with the volatile compounds in their bacteria as well. We also want to do a more robust analysis. And as I've been hinting at kind of throughout our results, we really want to go and get more samples. Our plan is to get more samples this spring, especially focusing on Toomey woodlot and getting more adults. And then we also want to try and get some environmental samples of places like under the cover boards where we're picking up the salamanders, because of the importance that we've seen, or that other researchers has seen, of the environment playing into what their microbiome is looking at, and the fact that we're getting very different like volatile profiles from Toomey versus Rose Lake. We want to see if maybe that environment is affecting the bacteria as well as the compounds associated with those salamanders from the different localities. And then moving on to long-term goals, of course, all depending on kind of what we find, and if these trends continue on with a larger sample size, we potentially would like to look at into doing things like behavior studies, exposing salamanders to chemical cues, and kind of seeing how they may react. All right, so just to wrap up, I would like to thank a lot of different people. So of course, I'd like to thank Dr. Mead and Dr. Whitaker, my mentors, who have been really helpful and wonderful, and definitely this project wouldn't have been possible without either of them. I'd also like to thank the Research Technology Support Facility here at MSU, and specifically Dr. Cassandra Johnny. She's been working with us very closely on the volatile analysis and showing us how to use all the new fancy software that we're not familiar with. I'd also like to thank the Scribner Labs, so Dr. Kim Scribner and Dr. Jeanette Kanefsky, who've allowed us to do all the DNA extractions in their lab. I'd also like to thank SparkNet for allowing us to use their cover boards for us gathering our samples, and of course Beacon for supporting and funding the project as well. Right, but thank you all for listening. Any questions? I'll try and monitor the chat as well.
Great job, Megan. If anybody has a question, please feel free to just unmute yourself, or you can use the chat box.
Hi. So I actually had a couple questions. First of all, I just want to say that talk was awesome. You just like quadrupled my knowledge of salamanders right there. So that was kind of cool. My first question was, so I understand that your two different like locations are like isolated geographically, but are they like distinct like physically? Like is the forest different in the two areas or anything like that?
Yeah, so they actually, yeah, so they are. So Rose Lake is kind of like a classic like forested area versus like Toomey. It of course is like forested, but right next to it is like a cattle pasture used by MSU. So we kind of think that maybe something with like, I don't know, like the pH or like the different soil nutrients or something that might be affecting it.
All right, cool. Yeah, thanks. And then, so the other thing I was wondering was and I think I caught this correctly. You said that the salamander skin is like selects for specific bacteria. Is that correct?
Yes.
Do you know how that like happens or how that like occurs?
Just based off of the research I've looked at, they just kind of believe it's kind of like any other environment where the salamanders just like their body or their skin specifically like favors just different bacteria to live there.
All right, cool. Yeah, thanks.
That
was a really cool talk.
Thank you.
Yeah.
Hi, Megan. Great talk. You powered through that very fast. It was great. Fast but comprehensively. So I was wondering, for the salamanders, did you collect the samples at the spot on the location, or did you like take them to the lab?
So we swabbed them in the field, but then we took them back just waiting for fecal pellets. And then at the end, we just released them where they were found.
Okay. And did you record how long it took for them to produce fecal pellets after you brought them back?
We did not. And Dr. Mead, she's been working with that. So maybe she could tell you a little bit more about that average time.
Yeah, so in general, we didn't want to keep the salamanders in captivity for too long. So usually within 24 to 48 hours. And if they didn't produce a fecal pellet by that point, we put them back into the field.
Okay, because I was wondering, based on the, I think it was Loudon, the graph by Loudon, when they stay in, I think if I interpreted it correctly, when they stay in captivity, their microbiome changes. So I was wondering if that would affect, yeah. Okay, thanks. I guess another question was that, so we talked about the forests being distinct in terms of the habitat, but I know on the map, they're obviously probably close to each other in the grand scheme of things, but do they have weather differences, for example? Would there be a reason if the northern tuni, or if that's tumi, whatever the name was, I forgot, if that was more northern, for example, and I don't know if it differs by a few days, whether that could result in more salamanders being young or not. I don't know if that's actually a thing that could happen.
Yeah, I would say that just overall, they probably have very similar weather patterns, probably because they're just like pretty close like together. We kind of think we got a lot of the juveniles because that was a lot of the Toomey samples were from like our first initial sampling sessions. And that was kind of earlier in the season when there were a lot more juveniles out.
Okay, that makes sense. Thanks.
Yeah, Megan, if you don't mind me butting in, another thing I would like to get more data on is it looks like there might be a seasonal effect because there was an effective date on the volatile composition. So, but we just didn't have enough data to really look at that yet. Any other questions? All right, well, Megan, you did a fantastic job. Let's all thank Megan again.
Yeah, great job, Megan.
Thank you all for coming.
Oh, and I do have friends here at the cottage in Maine who are also clapping for you. Yeah, so I will be posting this on our YouTube channel. So Megan, please feel free to share it anywhere you like. And of course, other Beaconites will have access to that as well. All right, well,
great.
If there's nothing else, I hope everyone has a great weekend. And we'll see you soon. This is our last Beacon seminar for the semester. So we'll be working on filling up the next round soon. All right. Bye, everyone.
Okay, bye.